for giving us this opportunity to represent lipid excipients at this prestigious forum of Indian Pharmaceutical Congress 73rd at the Hyderabad. Can I have my presentation, please? So, just further to a very enticing talk from Mr. Ravleen Singh on excipients and the regulations which are required. This talk will cover about lipids, lipid excipients, and what are the considerations that one needs to give, or what are the considerations that one needs to highlight when you are developing a lipid-based formulation systems. Next slide, please. Next slide, please. So if you see lipid-based drug delivery systems, they offer a very versatile applications. They can be used in for developing oral drug delivery systems, and the applications run from enhancing the bioavailability of poorly water-soluble APIs, developing sustained release formulations, developing taste tablets wherein they can be used as lubricants and also as taste masking agents. When you're developing topical drug delivery systems, they can be used as emulsifiers, penetration enhancers, and also as thickeners. They can be used in rectal and vaginal drug delivery system, where the hard fats offer very good tolerance to the rectal and vaginal mucosa. And also they are used very widely for developing injectable drug deliveries, wherein they are used as solubilizers, solvents, and also for developing liposome kind of drug deliveries. Next slide, please. So before we move on in this presentation, let us quickly understand the chemistry of these excipients, because they are niche. They certain consideration needs to be given, and so before that we will understand quickly on the chemistry of these products. Now, why oleochemistry? Because the Latin word for oils is oleo, and hence the chemistry of the oils or oleochemistry. Next slide, please. So basically lipids, they are naturally occurring compounds. They are insoluble in water, and they have certain solubilities in the organic solvents. So these lipids are basically classified as simple lipids and complex lipids. Simple lipids are our daily dietary lipids which we consume in our diet, like the butter, oil, ghee. They fall under the category of simple lipids, while complex lipids are prostaglandins, sphingolipids, or phospholipids, which are used for development of liposomes. May it be simple lipids or complex lipids, the basic building block behind these excipients are the fatty acids. Next slide, please. So the fatty acids, they are monocarboxylic acids chain followed by an acyl group. Now the number of carbon atoms that are linked to one another in a fatty acid chain determines the physicochemical property of the fatty acid. Also, the presence or absence of unsaturation or saturation also determines the physicochemical property. For example, if you see a fatty acid with carbon atoms ranging between C2 to C6, these fatty acids are water miscible. A classic example of such fatty acids is acetic acid, which we all have worked during our chemistry lab, and acetic acid is highly miscible in water. As the number of carbon atoms increases, the miscibility or solubility of the fatty acids in water decreases. So as the number of carbon atoms increases from C18 to C12, these fatty acids are medium chain fatty acids, and these are water miscible. So water dispersible. So they will form a milky dispersion in water. They will not separate out completely like when you mix oil and water, but at the same time, they will not form a homogeneous one-phase system. And these fatty acids are termed as medium chain fatty acids. Now, as further the number of carbon atoms increases, the solubility further decreases, and so when you have fatty acids ranging between C14 and above, these fatty acids are completely immiscible. So if the fatty acid is liquid, it will form a separate layer like when you add oil to water, and if they are wax, they will just settle down at the bottom of the beaker and they will not solubilize in water at all. Next slide, please. So let us see some properties of these fatty acids and how the physicochemical property is determined for them. So let us take the example of lauric acid and palmitic acid. Now lauric acid has 12 carbons in its chain length. It is a saturated fatty acid with no double bond. Palmitic acid is also a saturated fatty acid with 16 carbons in its chain length. Lauric acid is liquid in its physical state, whereas palmitic acid is a wax. So as the number of carbon atoms increases, the melting point of the fatty acid increases. Now let us take the example of stearic acid, oleic acid and linoleic acid. All these three fatty acids have 18 carbons in its chain length. Stearic acid has no unsaturation, that is, it's a saturated fatty acid. Linoleic and oleic acid have one unsaturation and two unsaturation, that is, one double bond and two double bond respectively in its chain length. So if you see the physical state of these three fatty acids, stearic acid is a wax, oleic and linoleic acid on the other hand are liquids. So presence or absence of saturation or unsaturation and also the number of carbon atoms in the chain length determines the physicochemical property of the fatty acids. Next slide please. So these lipid excipients which are available in the market, they are all esters of fatty acid and alcohol. So majorly these are manufactured by esterification reaction. So it could be a direct esterification reaction between an alcohol and the fatty acid, or they could be manufactured by interesterification. That is, two esters can be taken and they could be reacted together, or they could be manufactured by alcoholysis. That is, you take a long vegetable oil and you break it down into smaller components by means of an alcohol. Now these are derived, the fatty acids in the lipid excipients, they are derived from either vegetable or animal origin, and they are amphiphilic in nature because they have both the hydrophilic component which is contributed by the alcohol and the lipophilic component which is contributed by the fatty acid. Now depending upon the size of the alcohol molecule and the size of the fatty acid molecule, the corresponding HLB value and the application of the lipid excipient will be determined. Next slide please. So majorly all of these lipid excipients, they are a mixture of mono, di and triglycerides, and these mono, di and triglycerides are also determined by the extent of esterification reaction. That is, if the esterification reaction is conducted for a shorter duration, you will get majority of monoglycerides. So monoglycerides are what? Like if you say an example of glycerol, which is a trihydric alcohol, amongst the three hydroxyl group, one hydroxyl group would be replaced by a fatty acid, and you still have two hydroxyl group in the chain which contributes to the hydrophilicity of the molecule. This is followed by diglyceride where only one hydroxyl group is present, and this is in case of triglyceride all the three hydroxyl groups are replaced by the fatty acid, so that is the most lipophilic amongst the three. So all these lipid excipients, they are a mixture of mono, di and triglyceride, and depending upon the percentage of each of this component, again the corresponding HLB value and the application of the excipient will get determined. Next slide please. So these lipid excipients, they are always graded on an HLB scale. So HLB scale is hydrophilic lipophilic balance, and when the lipid excipients has an HLB ranging between 1 to 10, they are lipophilic, and when they have a HLB ranging between 11 to 20, they are hydrophilic in nature. Next slide please. Now, as we have seen in the earlier, during the previous slides, that the simple lipids, they are prone to digestion. So we all, human, irrespective of the race and ethnicity, we all consume oils, fats, butter in our daily diet, and hence a human body has an established system to digest these lipids, to cause emulsification of the lipids. And this digestion process is also applicable to the lipid excipients. So lipid excipients also undergo digestion, and this determines the performance of the lipid-based drug delivery system. Hence, it is very important that we consider this digestion process and how it affects the lipid-based excipients as well. This will also help us to establish a good in vitro, in vivo correlation when you are developing a lipid-based formulation. Next slide, please. So why should you consider a lipid-based formulation? Because you already have an established digestion system which will digest the excipient, and hence they render these excipients non-toxic. The physiological pathways are already established for their uptake, And they also protect the drug from precipitation, because on digestion they form micelles and the API remains solubilized in the GI fluid until it is absorbed by the intestinal cells. And also the surfactants which are used in these formulations, they help in improving the permeation of the API across the intestinal cells. Next slide, please. So, when you consume a diet containing lipid, in the stomach, they are acted upon by the gastric lipases, and the triglycerides or the larger molecular weight components are broken down into smaller components by the gastric lipases. And then as they enter into the intestine section, by natural body mechanism, the bile salts and pancreatic juices are secreted, and these are our natural body enzymes or natural body surfactants which cause digestion or emulsification of the lipids. And the lipid excipients also undergo the same pathway. Next slide, please. So in stomach, the triglycerides, which are the larger molecular weight components, they are broken down into diglycerides, monoglycerides, and free fatty acids. And the diglycerides are also broken down into further smaller components. In case of intestine, the PEG esters, they are partially digested, releasing the alcohol and the free fatty acid. And then when your API has solubility or affinity towards these components, it finds an easy entry inside through the intestinal cell wall. Next slide, please. So there is always a question like, when should you consume the lipid-based formulation, whether fed state is good or a fasted state is good. Also, there are certain cases where you find a good bioavailability or the bio studies are passing in the fasted state, but they're failing in the fed state. So let us understand why this phenomenon occurs. So in case of stomach, in fasted condition, the pH of the gastric fluid is between 1 to 2 and the stomach transit time is about 2 hours. But when food appears in stomach, the food buffers the pH of the stomach or the pH of the gastric fluid, and it increases to about 5. Also, the gastric transit time increases to about 3 to 4 hours. Now, because of the appearance of food or in the fed state, the lipid excipient is more exposed to the gastric lipases, more breaking down of the larger molecules take place, and this results in better digestion or better emulsification of the lipid excipient. Also in intestine, there is no much difference between the fed and the fasted state. The pH of intestine in both the cases is between 5 to 6, and also the intestinal transit time is about 4 hours to 5 hours. But when you consume food by natural body mechanism, bile salts and pancreatic juices are secreted, which causes emulsification of the lipid excipient. And because of this emulsification, either it will improve the solubility of the drug, giving you supra results, or it will result in precipitation of the API, giving you results which are below or lower than what you observe in the fasted state. Hence, this particular consideration should always be given when you're working with lipid excipients. Next slide, please. So when you are working on lipid-based drug delivery systems, if your API has an affinity towards a medium chain fatty acid or medium chain triglyceride, in that case, the API is always absorbed through the direct systemic circulation, because the molecular weight of the MCTs is lower, and hence they are absorbed from the systemic circulation, and the same pathway is followed by the API. Whereas if your API is highly lipophilic and it shows affinity towards long chain fatty acids, then long chain fatty acids, owing to their higher molecular weight, cannot enter into direct systemic circulation. They enter into the lacteal systems, and hence you can avoid the first pass metabolism. So there is a myth that lipid excipients can, if you use lipid excipients, you can avoid first pass metabolism, but that is not the case. Your API should be lipophilic and should show affinity towards long chain fatty acids, and only then you will be able to bypass the first pass metabolism. Next slide, please. So there is a consortium, Lipid Formulation Consortium, which has classified the lipid-based drug delivery systems into 4 types. Type 1 systems are the one wherein the API is highly lipophilic. It shows no affinity towards any surfactant. So it is only solubilized in the oil, filled into capsules, and is given to the patient. So the natural body mechanism or the natural surfactants like bile salts and pancreatic juices are responsible for emulsification of such systems. But the glitch for such excipients or for such systems is that there is a lot of intersubject variability, because the amount of bile salts and pancreatic juices which are secreted is quite variable from individual to individual. Hence, these systems are highly avoided and they are used only when the API shows no affinity towards any of the other surfactants or any other excipients. Then you have type 2 systems. So in type 2 systems, along with the oil, you also include a low HLV surfactant. And these systems, when they hydrate in the GI fluid, they emulsify. So these systems are self-emulsifying drug delivery systems or SED systems. And here the oil percentage is slightly lower than that of the type 1 systems. Since you're adding a known amount of surfactant to these systems, you can actually evaluate the performance of these systems and they don't rely on the natural body mechanisms. Then you have type 3 systems where again the oil concentration further reduces, and along with a low HLV surfactant you also include a high HLV surfactant and also a co-solvent. So this results in higher hydrophilicity or higher hydration of the system, and these, when they hydrate in the GI fluid, they form SMED, that is self micro emulsifying drug delivery systems. And then you have type 4 systems where there is no oil at all. You just have this, so these are usually formulated for polar drugs, which has got a log P value between 1 to 2. And these systems are just mixed with the surfactant and filled into capsule. And when they hydrate, they form micelles. And these micelles aid in improving the solubilization of the API in the GI fluid. Next slide, please. But whatever kind of systems you develop, it is very, very important that your API should have affinity towards this lipid, because then and only then these systems will be able to keep the API in solution. Otherwise, it will result in direct precipitation of the API and your whole effort of developing a lipid-based formulation will go waste. Next slide, please. Now, once you have developed a lipid-based formulation, it is a big challenge to predict their performance, because as we have seen in the previous slides, these excipients undergo digestion. So if you just rely on your in vitro dissolution test, this will not help you to give an accurate prediction of the performance of these excipients or of these formulations in vivo, because in vivo they are going to undergo digestion, they are going to undergo emulsification, and hence the correlation is difficult to establish if you just rely on simple plain in vitro dissolution testing. Next slide, please. So there are certain considerations which you need to take into account, like the volume of the dissolution medium. So usually when you develop a lipid-based formulation, they have or they exhibit a supersaturated state. Now what a supersaturated state is, they undergo hydration, and during hydration there is a saturated solution of the API which is formulated or developed in the vicinity of the formula. So in that case, if your surfactant or if your solubilizer is not enough, it may result in precipitation of the API also. So if you use a simple dissolution technique which has around 900 ml of dissolution medium, the sink condition will not actually predict what is happening to your formulation in vivo. Hence, it is important that you should consider the volume of the formulation and accordingly test the lipid-based drug delivery system. Also, the medium that is being used should mimic the body fluid. That is, it should be a biorelevant media, or it should consider the enzymes or the enzymatic action which takes place in the intestine. And also the hydrodynamics, because of the gastric motility, the hydrodynamics should be mimicked or should be simulated as that which are used, that which are there in the small intestine. Next, please. So, a recommendation is made to test your developed lipid-based formulation along with the regular dissolution testing. It is important that you also test your formulation using in vitro digestion test by pH stat. So here, what they do is they just take 36 ml of phosphate buffer of pH 6.5, and they add sodium taurodioxycholate and phosphatidylcholine, which mimics your in the bile salts and the pancreatic juices, and the solubilization capacity of your formulation is tested, and this helps to establish a good in vitro in vivo correlation for the formulation. Next slide, please. Apart from the testing and apart from evaluating the performance of the lipid based formulation, most of the lipid excipients which are available in the market, they comply to the USP EP Chinese pharmacopoeia, and also some of them comply to the Japanese pharmacopoeia. Most of the products are also have a food additive status. They have drug master files in major markets like US, Canada, Chinese CDMA. Then a lot of toxicological tests for the route of application, which is for which they are being used, are available. And a lot of market references are also available because these systems are there in market for some time now, and a lot of market references are also available. The manufacturing sites are ISO certified, GMP compliant, and also now considering the CSR approach, a lot of companies are giving emphasis on the RSPO certification. And because these excipients are derived from natural origin, it is relatively easier to get a RSPO certification, and the companies are working for this certification. Next slide, please. These are some of the market references which are available for lipid-based drug delivery systems, and you can see they are a good mix of oral, topical, as well as suppository systems. Next slide, please. So to conclude my talk, lipid formulations are derived from natural origin. They are functional excipients, that is, they exhibit a specific function in the formula. They can be used as bioavailability enhancers, or sustained release matrix forming agents, or as a lubricant, or a taste masking agent, or as an emulsifier in the topical formulation. They cover a wide range of application from oral, topical, for different routes of application. And since they are applicable to natural digestive pathways, they are digested. So hence they are a good system or a good means to ensure patient safety as well, provided your drug has an affinity for these excipients. So with this, I thank you for your attention, and I'm open for questions.
We had a series of meeting with Minister Mansukh Bhai Mandviya ji and Rajiv Raghuvanshi ji, who has, after deliberations with the IPAC, initiated this committee. Secondly, the IPAC guidelines are already there. So IPAC is headquartered in, it's a federation where we have our headquarters in USA. We have the branches in Europe, Japan, China. So already the IPAC standards are in place. And
one question, if we would like to formulate any formulations for cardiovascular, particularly these triglycerides have an impact on cholesterol. So how do you justify that using lipid formulations?
Understand, and yes, that is always a myth that, because they are lipids, they're going to add into your cholesterol content, or they're going to add into your fat content in the body. But no, because the amount of lipid excipients which are there in a dosage form is not to very high extent. It is in mini, you just consume one dose, or so the amount of excipient which is consumed in a dosage form is very less. And yes, correct. No, no, not at all, because they undergo digestion. Like I have detailed in the presentation, they undergo digestion and they're broken down and they are absorbed as well. So they do not cause, or they do not add into your cholesterol level. Cholesterol is completely, sorry? It is synthesized, but the molecular weight or the kind of structure which cholesterol has, these are just fatty acids. So, they will not add into your cholesterol manufacturing, or they will not add into your cholesterol content in the body. Okay.
topic what you discussed today is of vital importance for the formulation people, because a lot of things are happening when the excipients are concerned. In fact, whatever little time I had a discussion with him and was asking him how do we get out of this situation, because quite risky after what has happened recently in the international where we had exported some products and there were some very risky things. So this is of very vital importance, and I'm so happy that this subject is being discussed at length. Thank you so much. Thank you.
Thank you very much. Can you just have a